Hello everyone. I hope you all can hear me. Hello everyone. Hello to the host. I hope you all can hear me, please. Sorry if you can hear me. If I'm audible, can you please be raise a thumbs up so that I know that we all are on the same page. All right. Thank you so much. So, good day, everyone, to the host. I would like to welcome you all to the final day of Gogenebio. I am so excited for today because it has been 2 days of impact of training of pushing this forward, and I would like to specially say a big thank you to all the hosts worldwide, to the hosts and participants from Tunisia, from Philippines, from Nigeria, from Zambia, from Pakistan, from all over the world. Thank you for joining. Thank you for dedicating your time. Thank you for making this initiative a reality with us at Genoma Holdings, and we are so glad that this is a success already. So today is a live session, as we all know, and I would like to say a big thank you to all the participants worldwide. So thank you for your dedication to this vision. Thank you for dedicating yourself, for bringing that effort to the table. Thank you so much. We see all you do. We see your effort. We see the impact that has been made, and we are so grateful. And also, this is a call for us to delve deeper, to dig deep into the training that we've received, into what has been impacted to us. So, to all the hosts, to all the participants, I say a big thank you for your commitment, for your dedication, and for your hard work. Thank you so much. So, before we commence today's meeting, I would like to bring to the table um, our CEO, yes, the CEO and the founder of Genomark Holdings, the person of Mr. the person of Mr. Abraham Oluwase Olawale, as he gives us a lovely remark and a brief address on some very important things for the host, and then we start right away from there. So, you are welcome, sir. I think we need to wait for his. I don't know if he has trouble. Please let's wait as he comes up. So hello everyone, please. You can just wait for a while for Mr. Sheyolawale to join us before starting the question and answer. So, hello everyone. Mr. Sheyi Olawale is currently not on seat, so I think we can kickstart the live session for the question and answer right now. So, I would like to invite our dear director, the director of Genomark Holdings, the person of Mr. Sheon Agbola, the person who was the one that was actually um, it was one that prepared for the one section. So I would like to invite him now as he take us through the question and then answer the remarkable part of the questions as I round up. So Mr. Shion Adwola, you are kindly welcome to this meeting. I would like to hand the mic to you right now. So please, you can step on the podium. Mr. Shion, are you there? So while Mr. Sheung will be joining us in a few seconds, I would like to specially use this medium to tell us about Genoma codings. And since we are all aware about that, it's a good time to still refresh about Genoma. Okay, Mr. Shion is around. So, Mr. Shion, the director of Genoma Institute, the director of Genoma Institute, one of the subsidiaries in Genoma Holdings, will be taking us through day one question for Gogenebio. So please, can we send a thumbs up emoji or a love emoji as he comments on the questions, please? So Mr. Shion, you're kindly welcome and you can kickstart from here. We are already waiting. So thank you so much, everyone. I see that. Yeah. So Mr. Shion, you can kickstart with the questions, sir.
All right, thank you so much, Gloria. Good day, everyone. Please, can you hear me? I'd like to confirm if you can hear me.
Yes.
Hear me, sir.
Yes, we can hear you.
All right, thank you so much. Thank you. So I would like to firstly congratulate you all for the completion of this program. And I'd like to also appreciate our hosts across the region for, you know, taking up this initiative to host genomics and bioinformatics across, the regions. Thank you for helping us to advance our desire and our vision to ensure that genomics and bioinformatics education is spread across, you know, Africa, not just Africa alone, but is spread across, because we discover that bioinformatics, genomics and bioinformatics has been around for a while now. But then in this part of the world, in Africa especially, we have a research gap, we have knowledge gap as far as genomics and bioinformatics is concerned. And that's why Genomark Holdings is committed to ensuring that this gap is actually filled up. So thank you once again for this great initiative. So now I'm going to get right to the questions. I have a ton of questions here, and I hope that this session will be enough to answer the question. I've been able to go through some of the questions I have here. So I would like to say that, to make this clear first, before I begin to take some of the questions that I have here, I'd like to say that this is not actually a consultation session. So, because I saw that we have some question here that has to do with maybe someone carrying out research. And if you need consultation, we have a arm of Genomark Holdings that does consultation. So you can, reach out to them. You can go on our website, and you'll see that that's Genomark Services and Consults. You need consultation regarding your research. If you are carrying out a research and you need consultation regarding that, you could, just reach out to them for them to answer your questions. All right, so that's to say that I'm actually going to be omitting some questions. You would pardon me for that. All right, so let's get right to it. So the first question I have here is Joy from, I don't know which country, NCR. So here it says, can the software be used to design mutagenic primers with more than 20 base pair sequences? So definitely yes. Genius software can be used to design mutagenic primers. Now, mutagenic primers are primers beyond, the normal number of base pair that we have for primers. So primers are between 18 to 20 or 24, but mutagenic primers could be, longer than that. So yes, definitely we use it to design mutagenic primers. So it also depends on the length of the mutated region you are trying to target. Okay, so I think that's our question. So let's move to the next question. It says, does the primer design part can be doable to any modified system? Because we do genetic. Okay, I think this also has been answered. All right, so someone asked here, that's Jose. I don't know what region, said, is there a difference, advantage, disadvantage for using the prescribed web-based tools and Genius software as compared to using conda-based tools in terminal /bash? All right, so now I want us to understand that this particular initiative was a bit limited because bioinformatics is something that is broad. Please, if your mic is on, please can you just mute yourself? If your mic is on, please mute yourself. Thank you. Kindly mute yourself, please. Please mute yourself, Mr. Ahmed, please. Thank you.
Yawa.
Can you
Yawa. Ka gani. Hanya ba su kwana yamma wasu. Ai ko tafi ya san kwamfuta. Bambanci tafi ma kwamfuta ne.
your mom? Mr. Meg, kindly mute your mic, sir.
Yan kasuwan sun gina na
I have a question. If any one of you can answer it for me, please.
From their end. Since you are the one that studied this thing, you can mute the person.
Mister, please, hold on till we are done. Thank you so much.
Um, are you having any experience in core genome analysis?
I can't hear anyone speaking.
Same here. I can't hear anything as well.
Please kindly exercise patience. We are trying to sort the mic stuff.
We can't hear anything from this end too. We're just sitting here and just waiting for nothing.
Exercise patience. We are trying to sort the mic stuff.
From this end. Just waiting for nothing.
Hello, everyone. Please just exercise patience. We are trying to sort this disturbance we are experiencing from the people who have their mics on. So please just exercise patience for like few minutes. Should be sorted soon.
Hello, everyone. Please, I would like to know if you can hear me. Just drop a yes in the chat box. All right. Thank you. Thank you. Thank you. All right. So we want to apologize for the technical issues. We're just trying to ensure uh, we have some sanity here. Thank you. All right. So let's continue from where we stopped. So yeah, the question I have here is there a difference, advantage or disadvantage for using prescribed web-based tools and Genius software as compared to using Conda-based tools in Terminal slash Bash? Okay, so now, like I said earlier, this initiative is actually, um, is this initiative is to help those who are just finding their feet in genomics and bioinformatics. So you may have been hearing of genomics and bioinformatics for a while, and then you're a life scientist. Then if you want to know what genomics and bioinformatics is all about, and that's why we have the curriculum of this initiative built for those who are just getting to understand what genomics and bioinformatics is all about. So that's why you will see that it has a limit of to what we could explore. So to answer that question, I would say that definitely you could use other, you know, other tools. You can even use, the likes of R programming language to analyze your data. You can use Linux. You can use, different. Like I said, there are tons of web-based tools that you can use to analyze that you can use to make sense of your genomic data. So it's not just Genius software alone. We have tons of softwares. You have tons of web-based tools that you can use to, analyze your data. So yeah, I think that answers the question. So I'm going to move right to the next one. This is James from Philippines. Okay, it says, I'm grateful for the lifetime access. I was just wondering if there's an easy way for us to see differences between Genius 9.18 and whatever. Okay, so now for the Genius versions, so the reason why you're using the present version you're using is because we only have access to the present version. For the newest version, I mean the latest version, you have to pay a huge amount of money, to be able to have access to that, because the subscription is, way higher than the present one you're using. Yeah, and for the Genius software we are using presently, it could still perform some of the things the latest version could do. Maybe a slight difference. Okay, so yeah. Then we have Abdulaziz Okonlawo from Nigeria says, what are the differences between genetics and genomics? How is bioinformatics useful in cancer genomics? Okay, so there is a difference between genetics and genomics. Um, However, they actually interwoven, they are related, but there's a difference. So when you talk about genomics, just like we had we have it, explained in the session, genomics is when you're studying the entire DNA set of an organism. It could be bacteria, it could be plant, it could be animal. So you are trying to understand, you're trying to gain insight into the entire DNA set of the genomics of the genome of that particular organism. So that way you want to know what regions is, responsible for a particular condition. For example, maybe cancer. You want to know the genes that are expressed in a particular condition. Just like I said, cancer. You want to know the genes that are expressed, when um, during drought season in a plant. What are genes that are expressed during drought season? So that's just an example. Then genetics is, you are trying to understand, is the study of specific genes. Specific genes, it could be a couple of genes, the genes that are responsible for sickle cell, responsible for a particular disorder. So that's genetics. That's the difference between genetics and genomics. Genomics is the entire DNA set, the complete um, DNA set of that particular organism, while genetics is just, you know, a just a couple of genes, that you're trying to study. All right, so we have Titus from Abuja. Regarding NCBI, would it be right to say it has the same functionality with protein database, where you could get information on protein structures in his annotations and the rest? Yeah, again, I said this in the session that we have a number of databases. We don't just have NCBI, and ENA. We have Pepper Genome database. If you want to work on Pepper Genome, there's a database for that. So protein, we have protein database also, where you could get information on protein structures. Yes, definitely. That is right to say that. All right. Here this person says, are there IT internship or project opportunities available after the class? Okay, I will leave this to the marketing team to address this, because we still have a number of messages here. So at the end of this session, there will be um, information around other project opportunities that are going to be available, other internship opportunities that are going to be available, because it is one thing to have this knowledge, genomics and bioinformatics. It's another thing to be able to apply it to real world application. So the marketing team are going to go on to talk about that after the Q&A session. All right. So can I use the app on the phone? This is Cookie from Southern Nigerian. Can I use Nigeria? Can I use the app on the phone? I don't know what app you're talking about, but I want to assume you're talking about the Genius software. No, you can't use Genius software on the phone. All right. So, okay. Here is Stella from Port Harcourt. Says, could you please re-explain the following: how to download sequence files from NCBI database, importing to the Genius software, and annotating or identifying protein coding regions? Okay, I don't know if it is okay for me to go ahead to do this, but let me try and seek permission from the admin if we still have time to display this. All right. So I guess I may not be able to do that because we are really behind schedule right now. So, but what I would say is you could go ahead to watch the video. Just rewatch the video on how to download sequence files on NCBI. Just pause it and then try to understand what is being done. Okay. So then we have Abiga from Pakistan. When there are bioinformatics tools available like Primer designing, we can use Primer, okay, websites, more others. Then why do we have to use this Genius software? Kindly explain the specific reason for using Genius software to perform or to design Primer. Okay, so like I said, we definitely not going to use all the tools available on Genius in bioinformatics to explain how to design Primer for you. So we had to just make use of one to explain that. So, like I said, we have tons of web-based tools. We have tons of databases that we could use to explain, but there is no that time to do that. So, to answer Abiba's question, I would say that yeah, we have other tools that you can use to design your primer, and there is no any reason why we decided to use Genius. Because, yeah, other other web-based tools could also perform the same, you know, design just like we did on Genius software. So we just decided to use Genius software because we also wanted you to have access to Genius software. I mean, beyond just this designing primer on Genius software, you could also do some other analysis. You could do base calling. You could do, phylogenetic analysis also. Yeah, that's that. So we have Shegun from Ibadan. We have Shegun from Ibadan. Is bioinformatics applicable in cancer research and gene editing? Of course, bioinformatics is applicable in cancer research. And I think I've mentioned that earlier. Someone also answered that question. Bioinformatics has been able to help us to gain meaningful insight or make sense of our data. Now, beyond the sequence, many people have raw sequence. Many people have raw data they are working with or that they have, and they are yet to make sense of it. So, analyzing genomic data goes beyond the wet lab where you extract the DNA, you sequence it. So the question is, after you've sequenced it, what next? Your ATCG, your sequence is not going to scream out and just tell you, "Oh, I'm the one causing cancer." So this is where bioinformatics comes in. It helps us to be able to make sense of those sequence. Help us to tell. It helps us to identify those regions in the sequence that are responsible for whatever condition you are trying to look for. So definitely bioinformatics is applicable in cancer research. And as for gene editing, yes, it is. The insights we've been able to gain through bioinformatics has helped us to identify specific molecule, which we call molecular scissors, that we can use to, edit and reintroduce, genes, to modify certain defects like sickle cell, and some other conditions. So definitely there's a connection between bioinformatics and gene editing. So here's another question. Gracious Samuel from Nigeria. Can I install the software without a PC? And if so, how? You can install the software without a PC. You'll need your personal computer to be able to do that. Right. So we have Katrine from Port Harcourt. How will this program enhance my career capacity in developing more on bioinformatics and genomics? All right. So I would say that this is the first step. This is the first step of actually, developing yourself as far as bioinformatics is concerned. Bioinformatics is a very wide field, and if you are not careful, and you may get lost in that, in how shall I put it now? You could get lost in the complexity, like because this field of study doesn't sit still. I mean, it's evolving day after day. So it is your responsibility to, to keep up. And part of your responsibility is joining programs like this, looking forward to programs also that are beyond this, more advanced program that would actually help you. And not just programs, but programs that are related to your research interests. Because again, you don't want to be everywhere. You don't just want to be everywhere. You have to, niche down your interest to something, and that way you pursue it. So yeah, so I would just quickly say this: to enhance your career capacity, you should just look out for some of our programs, internship, virtual internship, where you carry out, where you engage with research based um, learning, and at the end of the day, you're able to come up with a capstone project. So yeah, you can be on the lookout for that. So you have Amit Chief um, from Christian, seeing Christian here. How to navigate Genius Hub. Yeah, so unfortunately, we're unable to really cover this in the session on how to navigate Genius Hub. But I will say that just keep, now that you have the Genius Hub, just keep using it. The more you use it, the more you get acquainted with it. So yeah, so don't just, after this program, don't just dump it. Ensure you use it, all, all the analysis that we've done so far. Try and practice, practice and practice and practice again. That way you'll be able to navigate it. All right, so I have Rosie here from Ambrose Ali University. Can those icons that weren't activated be activated? Yeah. So I know that there has been some issues around the plugins, not being, installed. Hello, please. I would like to know if you can still hear me. Can you still hear me, everyone? All right, thank you. So yeah, there's this issue regarding the plugins for Genius software, maybe one or two. Now, you may not necessarily need the last one that was not installed. However, if you still want to install it, it could be installed manually. You know, yeah, it could be installed manually. So, but it doesn't stop you from being able to perform any analysis you want to perform on Genius software. All right. So we have another question here. Wow, this is Edex from Edo State. Okay. This is Salahuddin. Yeah, sorry, Salahuddin from Edo State. Why the tutor was teaching, it talks about sickle cell, that there are treats for them. My question is he available in Nigeria? Okay, I'm not sure I said there was a treatment for sickle cell. I think I was talking about um, CRISPR, that's the molecular scissors, which could be used to edit. And I think that was actually performed on a particular patient. Yeah, if you check this the name of the patient, Olagere or something, yeah, who had sickle cell, and through what's it called, CRISPR gene editing, he was able to, his gene was edited and it was fine. It's fine and it's still currently alive. So I'm not sure this is available in Nigeria, and I think that's part of what I mentioned earlier, that in this part of the world, I mean, the research gap is really, really wide, really, really wide. So, and that's why I will commend the host for, coming up with this initiative to ensure we are closing in the gap little by little. Okay, so yeah, that's just answer to that. We have Precious from AAU Epoma. Some of the gigs on Genius application is inactive. How do we activate them? And there was a particular plugin that couldn't install. Okay, I think I've answered this earlier. Now, again, whenever you are installing your plugins, it is essential that your internet is stable. Your internet must be stable, because um, if your internet is not stable, you may not be able to, install some plugins. So some of those plugins are a bit heavier than the rest. So you will see some are 45 MB. So you need your internet to be stable to be able to install those plugins. So when you're having issue like that, you could just switch off your, internet connection and switch it on again to be able to fix that. So again, for the last plugin that was not installed, it doesn't really affect anything. You could still work. But like I said, you could, you could, what's it called? You could install it manually. Yeah. Okay. Thank you. So we have other questions here. Yeah, of course, you can get the class video. Yes, this is Adar from Tunisia. Okay, this is someone from South Africa. Genomic preceding data R software. Yes, I said you can use R to make sense of your data also. Is there another way to, this is Andre from Philippines, is there another way to perform functional annotation of genome aside using BVBRC? The website is quite buggy on my hand. Yes, there are other websites you can use to perform genome annotation. You can use Galaxy, yes, to perform that. You can also make use of Linux. You can make use of programming language to do that, but you will need to have a high capacity, as far as your computer is concerned. Yeah. So we have the reason why we opt for internet tools is because, you wouldn't need to have the stress of ensuring you have enough capacity on your computer. So that's why we just use BVPRC. And if you are getting started as a bioinformatician, it's not bad if you start with the internet tools before you proceed to using, the programming languages and or the command line. All right. So we have a lot of questions here. I hope I'll be able to. So I may just have to skip some that I've answered before. All right, so here it says, if I have genome Go data sets stored in VCF, can I extract 50K using Genome software? You may not be able to do that. You would not be able to do that. Yeah. So if you want to do that, you have to make use of tools like Blink. Yeah, you have to use Blink command line. You also have to use VCF tools itself. Okay. In the primer design example, how to choose a set of primers to amplify the whole system region equally, how to specific a region to be, okay, I'm not sure I understand this question, but let me just assume I do. So I would say that if you want to design your primer, definitely you already have a target region you want to use. So it is that region that you use to design your primer, and after it has been designed, just like it was shown in the video, the forward and the reverse, you export it from Genius software, and that is what you send to the lab. Yes, I think that now answers the question Farid was answering on the chat box. The primers we design are optimized for wet lab. Yes, they are. All right, so let's move on. While creating phylogenetic trees, I noticed percentage values on the branches and I didn't understand what exactly they indicate. Okay, so the percentage values are actually indicating similarity and dissimilarity. And so when you have, or you could refer to them as bootstrap value. So when you have a percentage of 70, 80, it shows that you have, similarity. When you have something lower, like 60 or 50, there's a bit of divergence. So that's basically what that is. And I think I showed the heat map on Genius software, how to see this percentage across the strains you're comparing. So that will give you like a glance of what, how related or how divergent the strains you're comparing are. Okay, so this is someone from Tunisia also. During your presentation, you didn't mention GFF files. You could please explain what they have, how they differ from FASTQ files. Okay. All right. So VCF files, like I said, we have different types of files. The only one we covered was FASTQ, GFF, and PAM. So your raw sequence, when you get your DNA sequence raw from the sequencing platform, comes in a FASTQ. Yeah. So when you map it to reference, the file that is generated is your BAM. And when you now identify the variant, that's, yeah, the mutation that's occurred, when you map it to the reference genome, it comes in the VCF file. So your variant, your VCF file is like the file that contains all the variants that you've identified. Okay, so that's basically what that means. Using genomic sequencing to identify AMR, is it possible for protein binding sites to keep changing for a particular drug, thereby causing recurrent AMR? Yes, of course, it is possible. It is part of the resistance mechanism. Like if you check the 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 antimicrobial resistance identification we did on BVBRC, you will see that we have classification, and that classification is also the same thing as a resistance mechanism. We've been able to identify what antibiotics class, what antibiotics they, they are resistant to, and what genes can fight. That classification help us to be able to understand the way they go about it. That will inform, you know, drug development, drug design. So definitely, yeah, it's possible for the protein binding sites to keep changing, and that could, cause recurrent, resistance, which is part of resistance mechanism. These bacteria they use, to resist, as a result of exposure to antibiotics and all that. Okay, so we have in low resource settings, what are the most practical genomic technologies that institutions should prioritize for disease surveillance and research? Okay, uh, well, I don't know. I think this is an issue. This is an issue for us, in this part of the world. Well, I would say maybe if, because the thing is we have there is no, how should I put it now? There is no genomics technologies that is not expensive. I mean, maybe they are more expensive than each other, yeah, but I think you may just have to start from somewhere, maybe having your PCR, and all. Then I would say that if you could apply for grants, if you have a research you're working from, this is Patrick from Indula. So I'm saying Patrick from Indula. If you have a research you're working on, if you could apply for grants, that may be able to afford you um, to get some of this technology that you could use to, sequence and all. All right, someone asks, do we have this program at master's level? Okay, no, no. Do you offer scholarships? Yes, we offer scholarships, and our marketing team, they're going to talk more on that. So someone is asking, what is genomes from Zambia? I think we've answered that already. Okay, I'm just going to skip some questions I've answered before. Okay, this is Isa Ali from Sokoto. Isa Ali from Sokoto. Please, could you enlighten me more what the use of BLAST? What the use of BLAST is for? Okay, BLAST, as it is called, it is basic local alignment search tool. Let me just say a little. Let me just say something. Now, I noticed that this even happened for professionals when they sequence their data. It could be just partial CDS, maybe 16S, and they want to carry out molecular identification. You realize that after the organization that performed the sequencing is done and the organization send the file to them, what they do is just they go on NCBI and just dump the file on NCBI for submission. It doesn't work like that. Or they just submit it for blasting for identification. Yeah, you may have had a prior knowledge of what the the sequence you're working with is, but you also want to identify it, making use of blast just for credibility and accuracy. So what most people do is they just dump it on NCPI for blasting and then it generates error. So when it comes to blasting requires some fundamental step, and part of that fundamental step is base calling. And I remember that this was covered in transcriptomics class during quality control, that whenever you have your file generated, there are some errors, you know there are some errors that could be found in the sequence. So this is where quality control is needed. And if you're doing molecular identification, this is where base calling is also needed, because base calling will help you to assign the most likely nucleotide base in the region of the DNA sequence you're working with. And is after you're done with this, you'll be able to perform blasting. So blasting will help you match the sequence with other sequences on the database. So the ones that have the highest percentage identity will be generated, and then based on the parameters like percentage identity, you could have like hundreds of them showing up or just 100 showing up. So the one that has the highest percentage identity is what you're going to, you know, go for. Is going to be the name of the sequence you're working with. Okay, so we have how do you transfer a sequence into Genius software. So just watch the video again. Okay, I'll say you should watch the video again. Okay, so you can just watch the video again. There are lots of questions here. Let me check. Okay, here's another question from Jack from Ndola. How do bioinformaticians handle and analyze large data set? This is also a challenge. This is also a challenge, yeah, because there are some resource-intensive genomic data, like there are some resource-intensive data, so you will need high power capacity computer. So, well, you could start by using Linux. Just ensure that you have enough space in your computer if you're using Linux, because again, it may get to a point where you may not be able to further analyze your data. If, for example, you are working with metagenomics data are always very huge. So yeah, so I think it's a challenge. Okay, so but you can start with using Linux. You can start with using some internet tools. I think Use Galaxy could also, do that. It's just the only limitation is sometimes it could be very slow. Yeah, but if you could get access to, high power computer, that would help you to be able to, handle large data sets. All right, so we have Charity from Zambia. Okay, what type of method is used to convert, let's say, a blood sample into data that you can use on your computer? Okay, this is this question is actually interesting because um it gets a bit, maybe should I use the word spooky, when you're trying to imagine how does a liquid or a solution finally became a digital file. So I think this is the question Charity is trying to ask. So it starts from your molecular lab, where after you've collected your blood sample, you have to extract the DNA. So this is work of those who are working in the molecular lab. So you extract the DNA because every of this sample has DNA. Your saliva, the blood sample, you could extract the DNA from it. So Yeah. So once you've extracted the DNA, okay, I'm just going to explain this in a very, basic form. So once you've extracted the DNA from the blood sample, let's say you're trying to identify plasmodium, like malaria parasite, from the blood sample of the patient, and you've collected the blood sample, you need to extract the DNA. After you've extracted the DNA, then you proceed to, you know, PCR. Okay. So now this technology, just like I said, genomic technologies are what help us, in a basic explanation again, help us to be able to convert this DNA to a digital file, like your Illumina helps you to, now there are a lot of complexity to this, but that may not really help you at the moment. So, but those genomics technology are what help us to be able to convert this, blood sample, the DNA that we extracted from the blood sample, into digital file that you now have, that you could see, your ATCG and all. I don't know if that answers your question. Okay, so we have Adedeji Olusegun from Oshogbo. In gene annotation before primer design that will eventually lead to PCR, how do you know the specific region you're actually looking for in the DNA sequence? Okay, so of course, you don't just design primer out of the blues, and you have to design primer based on your research. So if you are carrying out a research, and part of the research objective is you're trying to identify a particular region, a region has been a region of interest to you, and then you want to amplify it for further studies. So this is how you get to know what region. So let's say you're working with a particular microorganism, and then you've identified, based on your research, of course you must have done literature review, and you already know what region you want to work with. And when you have that data on Genius software or any tools you want to use, then you will be able to identify, based on the literature review you have done, you already know the region you are supposed to target. So just like you will see that the regions were displayed on Genius software, so is that region that you map to be able to, design your primer. Okay, so that's how you go about that. But you don't just design primer out of the blues. You must have a target region. Those, it could be a gene that confers resistance. It could be a gene that confers what trait you're looking for. All right, so we have a guy from Southern Africa, that's Zambia. How best can the genomics be used in food lab? Okay, yeah, it could be used in so many ways. I mean, genomics could be used in so many ways. Food pathogen. We have food pathogen also. So it could be used. You could sequence that, the isolate you got from the food. You could sequence that to understand what virulence factor they they exhibit. The toxins and all that. So yeah. So we do not have much time, so I'm going to rush through the rest of the question. We have Karen from Zambia. I want to know more about the course. What does he? Okay, okay. I'm not sure I get this question very well. Okay, we have Abdusalam from Osun State. Is bioinformatics mainly used to get information or to document a research? Okay. Now, let me just explain that again, and I've mentioned that earlier. Bioinformatics is what helps us to be able to make sense of genomic data. Like I said, it goes beyond just having your data, your sequence. It is bioinformatics through the use of internet tools, the use of software, the use of programming language. You are able to make sense of that data, to analyze your data. Whatever you're looking for, every, what's it called now? Every internet tools, every what's it called, programming language has packages that could help you, you know, unpack those things in your sequence. So yeah, it's not just only to get information, but the information that you get are also expected to be interpreted. Hello, please, can you still hear me? All right, thank you. So yeah, we have two more questions here. Two more questions. Okay, everything about genomics and bioinformatics. Okay, so um, yeah, we have, do I require a prior data analysis knowledge to be able to use the data generated and analyze in this training for personal work? Okay. What I would say is, if you are already finding yourself in the field of bioinformatics, you're definitely going to analyze data. So you don't have to necessarily have, you know, knowledge about data analysis before you could, before you could begin to analyze data, genomic data in that sense. Maybe having a data analysis knowledge could, contribute. It could contribute to it, but you don't necessarily have to have a data analysis knowledge before you begin to do that. All right, so someone says here everything about genomics and bioinformatics, that's a DM on. So I don't know what you mean by that, but what I would say is you can't know everything about genomics and bioinformatics. You have to start from somewhere. Like I said, you have to just start from somewhere. Just start from somewhere. Start from the fundamentals, because the fundamental knowledge is very important to be able to begin to come up with innovation, to be able to come up with ideas relating to research. These fundamental aspects are what is going to be a launching pad for you, are what is going to spur, you know, research ideas, research interests. So you can't necessarily know, you can't possibly know bioinformatics just within, 3 days. So this is just to, help you put your feet in the waters, so that you can begin your exploration. So, and that's what you've started. You're already on the right path. So yeah, so just, I would say, just continue to look out for research based training. Yeah, I would encourage research based training, because I would emphasize that again. Yeah, having all this knowledge is not enough. You need to be able to apply them. You need to be able to practice. You need to be able to do the hands-on. And that's why you would see that we had to shorten the theoretical aspect. As much as the theoretical aspect is necessary, we had to shorten that, because we also realize that the hands-on is very important. I mean, maybe you may not be able to understand some theoretical aspect, but the truth is, the moment you begin to practice, the moment you begin to go on with the hands-on, the theoretical aspect will catch on later. Because while you're trying to, analyze your data, while you're trying to carry out the practical aspect, there are some questions that will be popping up, and that way it will lead you to, doing more research, reading up more to understand some of those concepts in genomics and bioinformatics. So yeah, so that's what I would say. So I will just quickly go through some questions now. If I'm omitting some important question, just go through that now, and also check the chat box if there are some questions on the chat box.
Mr. Shion, we can't hear you, sir.
Yeah, I'm still here. I'm trying to go through some of the questions if I'm not omitting any. All right. I think that will be all from my end. So I think there's a question here in the chat box. Hi, Dr. Assam from Pakistan. I would like to appreciate. Okay, I think this is just a comment. I request to please also introduce the course data analysis using. Okay, that's not a question. Okay, I think it will be a wrap from my end. Thank you, everyone. I hope I was able to answer all your questions. Yeah, thank you.
Thank you so much, Mr. Our director, Shion. Thank you so much. I mean, your wealth of knowledge on bioinformatics is truly, truly impressive. Thank you so much. And now we'll be bringing to the call Mr. Sorry, give me a few seconds. All right. Okay, all right, everyone. We'll be delving into the questions soon. And before going there, we'd like to bring to the call our founder and the CEO of Genomark Holdings, the person of Mr. Oluwaseyi Olade Abraham, and joins us and gives us some information on Genomark Holdings as regarding the marketing aspect. So, Mr. Sheyi, you are welcome to the call. If you are there, you can pick it up from here. Mr. Sheyi, okay, I don't think he's on the call right now. Okay. Okay, so while we are waiting for Mr. Olawale, I would like the host for Gogenebio to please send the link for suggestion and clarification, no, suggestion and feedback to all our participants worldwide. It is very important to know review from Gogenebio for day one and day two. So we'd like to hear back from you. We'd like to know what your feedbacks are. So please, we'd like you to use the link that will be sent to you, that will be sent to your group. So please use that and then let us know what your opinions are. Okay, what do you learn or how was Gojen Bio for you? How impactful has it been for your community and what you intend to do with it? So that would be that for the link. And also Mrs. For day two questions, Mr. Shion, the director for Genomic Institute, has taken us through the day one question. So for day two questions, Miss Musraya, the team lead for GoGen Bio, yes. and the team lead for one of our subsidiary at Genmark Holdings will be taking us through the day 2 session. But before we do that, I would like to, I think Mr. Olawale, the founder and the CEO, of Genmark Holdings is on the group, so he's on the call right now. So I would like him to pick it up from here. Mr. Sheyi, you are welcome, sir. Mr. Sheyi, you can pick it up from here.
Amazing, amazing, amazing. Thank you so much for having me.
Yes,
Please,
sir.
can you hear me? Yeah.
Yes, sir. We can hear you, sir. We can hear you.
All
Welcome,
right,
sir.
right, all right. So, I'm so happy to be here. I've quite been seeing some amazing capacity building and building of competence that has been going on across different regions around the world. Um, I can see Howard Mercado. I believe you're from the Philippines, right? Howard Mercado. Yeah, yeah, from the Philippines. Yeah. And I want to believe we have quite a number of different participants from the Philippines, from Zambia, from okay, I think Promise Paul want to say something. Promise Paul. Do you want to say something, or you just indicating that you are from Philippines? Or yeah. So we have those from Tunisia. All right. We have people from Nigeria. I mean, from over 33 locations that hosted Gogen Bio. And that is really, really tremendous and impactful driven for us, and also purposeful field, because that is what Genomark has come to do. That is what we are after, such that we're able to build the next generation of scientists out there that could actually drive uh, that could actually drive you know, innovations that would actually drive development around the world, right? Contributing to the scientific community, right? And paving the way for the forthcoming generation. And that is why we're doing this. And we understand that genomics and bioinformatics is an integral part of all biotech or biological related space or fields or domains, right? Genomics and bioinformatics is a very, very integral part. I mean, we have people who are in the medical field, they still want to do something in genomics because it has something to do with um, it has something to do with precision medicine, right? Diagnosis, genetic diagnostics. And the field is evolving, right? There's a lot of disruption that is taking place, a lot of innovations that is taking place. And now in the era of AI, it is important that we understand that the revolution, right, will happen very fast. And we must be in the midst of it, right? We have to be in the midst of it and to see that we are able to also contribute and see that we are relevant, right, from time to time. And that is why we are doing all of these things. And I want to say, I believe that the committee of the Gogen Bio at Genomark has also relayed this, but I just want to quickly shift this in. We're looking forward to see that we have subsequent Gogen Bio initiative. This is the first one, and I want to say it's really, really successful, isn't it? Kind of can I just get some replies, maybe emojis? Come on, guys. I hope you guys like learn from it. And yeah, it's yeah, yeah, yeah. Thank you, Howard. I can see Omolade. I can see, yeah. Thank you, guys. Thank you, guys. Yeah, yeah. And I want to believe that you want more of this, right? You want more of this. You want more of this kind of impact driven initiative to bring to be brought to your environment, right? And to see that we're able to drive more impact together, right? We want to involve everyone. We want to involve you. I want to see that we're able to achieve quite the same thing with you guys, right? And that is why we are here. I mean, look at the Genius software. We sell this Genius software. If you want to go online to get this Genius software for a student, is like $200. You want to get it for institution or you being as a professor, you have to pay like $1,000. But we have to push to make sure that such I mean that Genius software is available for you guys for free to be able to access. I mean, that's a lot, right? And to me, I feel like it's only God that can bless us, right? God should bless us, right? Oh yeah, so our reward is in heaven. But at the same time, we're just, I mean, our reward is the fact that you get trained, you have the competence, right? And you are able to build on it. So please don't stop there. That's my advice. Don't stop there, right? Build on what you have already. Build it. Keep working on it. Yeah, keep trying to push, right? Keep trying to learn. Prepare yourself, because there's a lot of job opportunity out there in genomics and bioinformatics. A lot of institutions have not begun to incorporate it. It's an emerging field. It means that it's going to industries, biotech, startup, academias will begin to incorporate these things into their systems, into their operations. We're going to be seeing more precision medicine startup, the genetic test and diagnosis. I mean, the future is revolutionizing, right? It's. I mean, there's a lot of things that would take place in the coming days, right? That would require people who have already positioned themselves in the field, who have built competence, waiting for the available opportunity in the nearest future as it arrives. So while you build yourself, when it comes, it funds you, and it's going to be a game changing for you, right? A very, very game changing for you. So thank you so much for collaborating with us. And I want to say, let's look forward to the next one, the next Gogenebio coming up in maybe three to 4 months time, or let's say 3 months time or 4 months time, right? We are looking forward to have it maybe subsequent time in a year, maybe like 3 or 4 times if possible in a year. Meanwhile, we're also looking forward to funding. We did this from Genomark pockets, right, and all of that, right? So it's really not that easy, but I mean, we did it, right? We did it, and we don't want to stop there. So also, we're looking forward to a form of collaboration. So if you have people, partners who want to collaborate on this Gogenebio, to reach out to people all over the world to train them and all of that, build competence and set them ready. Even in the fourth Gogenebio, we want to see how we can incorporate research initiatives building in those areas as well, right? Especially in the area of innovation and all. So it's very, very key. So we look forward to that. So in case you have any body or any organizations that you think would want to partner with this kind of initiative, just reach out to the committee and just let us know, or maybe you can just let us know on the group, or reach out to your host. Your host will reach out to us so that we'll be able to do. But meanwhile, our host, we are looking forward to still hosting this particular initiative with you going forward. All right. So the last thing I want to say is this. By the way, thank you so much. So the last thing I want to say very quickly, I think I have my slide here. I just want to present. We have a form of an opportunity for you guys. Let me just open my slide and present it. Yeah, I'm working on my slide. It's something I really want to present to you guys or show you. So kindly let me know the moment you can see my screen. Please, can you see my screen? Yeah. Okay. Very good. Very good. All right. So this is it. With the hope of continuing this impact initiatives, we are actually proposing a virtual research internship program. Okay, this is supposed to be a virtual research internship program. Okay, so just give me a minute while I just quickly do this. Okay, so with the hope that we need to continue building this competence apart from the forthcoming Gogenebio that will take place in months, an interval within the year, right? We actually proposing a virtual research internship program that is going to take you from a virtual, research beginner right, to an expert in the research space as far as genomics and bioinformatics is concerned. So basically, I would like to say that it is not enough to say that you have skill, you are able to analyze the data. It is also important you are able to have a knowledge that would enable you to be able to interpret this data, right, into a research significance, and that would enable you to be able to really be able to interpret your work and then be able to pull a research paper together, right, from all the results you're going to get as part of the bioinformatics analysis on a specific research topic, research aims and objective is concerned, right? So in that way, you are able to really work with a team. Like that's the essence of this research internship program. You'll be able to work with a team from around the world, right? As a group, and then you are able to carry out, draft a research objective, research aim and objective across different domains of interest in genomics and bioinformatics, right? Either you want to apply it to humans, animal, plants, or microbial. We have all of that in place. We also have capabilities as touching innovation tools like bioinformatics, innovations in biotech through genomics and bioinformatics, more like building app, data science, being able to work with AI, machine learning, and also being able to build bioinformatics tools that you can deploy. I mean, there's quite a number of different institutions out there today that really are looking for people who have the knowledge and experience beyond just bioinformatics analysis, but also ability to be able to develop tools, right? Bioinformatics tools or genomic tools that could analyze data. I mean, there are a lot of emerging problems coming up in the near future that the present tools that we have are unable to do that, but with the help of machine learning and models, algorithms, we have seen that quite a number of different innovations is taking place rapidly, and you can actually dive into that field as well. So now that you have a basic understanding or some level of basic to intermediate understanding of how you can maneuver or dive in and dissect genomics and bioinformatics, it is also important that you are able to gain real life research experience in that area to add to what you have. Are you getting what I'm trying to say? And that's the essence of this program. So in essence, in this research internship program that I would like to introduce to you guys is going to take you through expert mentorship. So there's going to be once in a week live session with dedicated mentor. And this particular aspect is worth like $5,000 because we're bringing these mentors on board. And then definitely we're going to be, paying them. We're going to be working with them. We're going to be, how do I say, compensate them rather in quite a number of different ways. That's the word, right? And these mentors are going to be working with you for like 8 weeks rather. It's going to be an 8-week internship. Then there's going to be training and development that you need for opportunities in your career, right? And then you are going to be able to access an automated research platform called Omics Board. For any research program to access on Omics Board costs $1,000, right? For any research you want to access on Omics Board, it costs $1,000, but we are going to make it free. So it's a very robust platform that takes you from beginner to advanced, and it's a step-by-step research classes on Omics Board because it's self-paid space. So while you have weekly session with mentor, you have a whole 7 days in a week access to specific modules that you need to watch, and then you will need to apply that particular module to a research objective, and then you are able to do the research together with your team on. So it's more like killing 3 birds with 1 stone, right? 1, you learn the skill set. 2, you learn the research experience. 3, you do the research, real life on a real life data with your team, and then you are able to craft a specific project at the end of the day. Now, there's a high possibility that such project could be published depending on the quality of the paperwork that you do. And you get what I'm trying to say. There are quite a number of different research papers that are currently being working on to publish. And I think just now I got information from a journal who were willing to actually publish some of our papers very, very well. And you get what I'm trying to say. So that is very, very key, you see. And we are actually willing to see that we're able to work with you guys from around the world to see that we're able to build your research experience, research skill set, and at the end of the day, you have a very quality project that could be published depending on the content and our outcome. Are you get what I'm trying to say? And also, you're going to get access to this platform for free. Then, because this internship is a scholarship based internship, right? Then we have skill advancement that keeps you at the top wherever you find yourself, right? And then research project completion and publication, what I just said, depending on the quality of that work, right? Perfect beginner experience as well. So these are a lot of bonuses. You're going to get recommendation letter for pursuing your masters and PhD. We have seen people who have traveled out, who went to do their masters, PhD in US, UK, China, quite a number of different places actually that they went. I mean, they won this scholarship. They won a funding for their PhD, and they get recommendation letter from us. They did internship with us, and it actually gave them that opportunity. Then we have certificate that opens the door for you, connection with professionals all over the world, structured learning, on-demand training videos, group classes, hands-on session, mentorship. I mean, it's quite a number of robust experience, and you're going to be exploring real-time advanced bioinformatics project, okay? Real-time on real-life data, right? So I'm going to take you through overview of Omics board and just show you quickly how Omics board looks like. Is that okay? Omics board. So that is omicsboard.com, right? Please, I want to believe you can still see my screen, right? Please, can you still see my screen? Okay, great. So this is Omics board. You can just type Omics board on your phone right now, on your laptop, omicsboard.com, okay? Omics board from the word dashboard. So Omics and board, right? Omicsboard.com. So this is omicsboard.com. It's a place where you dive into omics-based research, transform your findings into publishable papers, gain in-demand skills and expertise as you explore, collaborate, and unlock the power of omics data, right? So these are some of the experiences or skill sets you are going to get access to. Recommendation letter, flexible duration, skills and expert development, personalized mentorship. Like I said, you're able to craft paper, work on real life paper data. So, but let me show you through the intense program. So these are the different kind of programs we have, that this platform have. So we are actually kind of collaborating, even though it's powered by Genomark, but this particular platform is owned by another company, right? And we are partnering with them to make sure that some of these programs are made available to our interns, right? So that is if you come on board with us as an intern. So we have cancer genomics, Parkinson's disease research, collector cancer research. You can see that. These are the research aim that is going to be conducted, conduct comprehensive genomic profiling and variant analysis across various cancer cell lines. We have Parkinson's disease, we have collector cancer, we have prostate cancer research, more like to study the gut and urinary microbiome of prostate cancer patients. Can you see that? Human microbiome for those who want to leverage or explore metagenomics, the community of microorganisms. And the essence of all of these things ultimately to develop target intervention for improved management and prevention of outbreak and disease, prevention of diseases, right? We have epidemiology and public health. This one has to do with outbreak. For those of you interested in something like COVID, outbreak, Ebola, and all of those things, monkeypox, this is very perfect for you, right? We have infectious disease, those who are so much interested in AMR, virulence factors, and all of those things. This is for virulence, this is for mobile gene elements. These are for comparative genomics, plant genomics, nutrigenomics. My God, we have anti-cancer. This is therapeutic, right? Where you are able to build, to explore various nature genetic material from the nature, from the environment, either from sea, from soil, from volcanic area, from hot spring. So we just like look from how we can get genetic materials from all of those hot regions, and then see how we can mine therapeutic peptide from them through genomics, proteomics, and also using machine learning. So this is a multi-omics research base. So can you see the research aim? To systematically identify and utilize biostatic genes from natural sources, employing multi-omics data and machine learning techniques to develop peptide-based anti-cancer therapeutics to improve precision and efficacy in plant treatment. There's quite a number of different ones. So, but let me just take you through the platform, you can just go to, I mean, this particular link will also be sent to you guys so that you can explore the program and see the available research internship program we have. So, so you can see this is the platform. It said embark on the profound exploration of genomics and bioinformatics, where you master essential skills, foster global collaboration, and translate your research into publishable discovery of significance and impact. Isn't it amazing? Yeah, isn't it amazing? These are program features, explore omics sciences, and some practical training sessions, skills, and expert development. I think I've said this earlier. These are the available programs here, actually, right? We should have plant genomics, please. So the other one I want to share is learn how to deploy a genomics data ML model. Please, how do I say it now? Please, we need to add a plant genomics here. Please, are there people who are so much interested in plants? Anybody? Can you just signify? Is there anybody who's so much interested in plants? Is there anybody who's interested in plants or plant research based? Anyone? Okay, so maybe not currently here, but maybe on YouTube we could have that. Yeah, yeah. Okay, so she said medicinal plants. Okay, that's impressive. That's impressive. So the other one I want to show you is learn how to deploy genomics data machine model. So you are going to master end-to-end process of applying machine learning genomics. So from cleaning data, preparing genomic data sets, evaluating and deploying models, you will learn how to turn complex data into meaningful predictions that power modern biotech innovation. Here in this other program here, you're going to learn how to create interactive bioinformatics tools in research and biotech projects. This add-on course guides you through building and deploying functional web app with Python and Streamline, making it easy to visualize, share, and interpret genomics and biological data in real time. So these are the available programs that we have. And I want to say that the worth of this particular program, it's priceless. I mean, it's really priceless. Yeah, I don't know if it's okay. These are some of the programs that said is available because I don't have much time again. So the worth of the program, I think, is over $3,000, right? Over $3,000, I think that is the worth of the entire program. But by the grace of God, I think I'm looking for something. Yes, so here it is. So it's starting by November, right? And it's going to run for 8 weeks. There's going to be a once in a week live session with mentor, and then you get access to the omics board for free, the entire program on omics board for free as well. So meanwhile, we usually ask for a $30 commitment fee. I repeat, we usually ask for a $30 commitment fee. The $30 does not amount to paying for this program because it's 8 weeks. That's 2 months. It's going to be a research base. You're going to get access to priceless things, tools, resources, data sets, and all of that. So, but the commitment fee, it is to call for your commitments to make sure that you're able to commit to this. That's the first thing. And number two is that we use part of that money also to see that we're able to compensate the mentors. That's number two. And at times, some of this, if you are able to proceed to having a very high quality paper, that at times we try to use funds from that to be able to address that as well, right? To at least some extent. Though we have global collaborators and platforms that we try to work with to be able to see that we push some of those projects, but only high quality based projects. Okay, that's it basically. So that means you have to put in the work to make sure that the work is quality. We have a research publication team in place that look into your work and to determine how quality it is and why you work with mentor to see how you can improve it as well. Are you getting what I'm trying to say? So, but I think because you guys are currently the current cohort of Gogenebio, you are going to, you have been given a more discounts. I think the committee proposed to give you guys more discounts in your in the application fee from $30 to $25, and with a deadline, actually with a deadline. So, but I just want to see is there I want to believe you guys will be interested to take in this initiative and take your skills forward beyond what you've currently gained or acquire. Am I right? Am I right? So can I see a reply very quickly? Can I see a reply very quickly? I want to believe that we have people who want to take this up, this research internship, this opportunity up to leverage the the $300 worth of research internship program in 2 months. Yes, yes, yes. Okay, very good, very good. Because I'm going now. So please, uh, by the through the courtesy of the committee of the Gogenebio, you are going to be getting $5 discount to pay $25. And also, this is research internship program is usually by selection. We don't usually give everyone. Are you get what I'm saying? So it's likely we review the application to see. Yeah, yeah, you can pay in installments. You can pay in installments as well. If you can pay in installment in case you don't have all of the money, you can pay in installments. Okay. So, but what I'm trying to say in essence is that we usually select people. We usually review applications to select who wins or who would actually take in the research application. But according to the partnership with the Cogen Bio Committee and Genemark Institute, they are actually, I mean, is it breaking the protocol now to give you guys a free slot, a free access to the program as many that are so interested to take it now. And the application ends, I think, latest by Tuesday. Yeah, I think the application ends latest by Tuesday. So in case you are interested, I would say that you start joining in. I don't know if the WhatsApp group has been sent across, yeah, to the groups. But I will request that you post the WhatsApp group on Google, I mean on YouTube chat room and also on Google Meet, the team, the committee. Please, can you do that? Since they already have free slots to the program directly. So please, can we have that? Can we have that so that they can start joining and hence they gain free access to the program? So please, I look forward to meeting you guys at the internship while we work together to build something meaningful and transform the scientific community through different research projects, aims and objectives, and as we work on real life data. And also as you take your skill set to a whole new level, okay, and get yourself prepared for global opportunities, scholarship jobs, and any other kind of paid internship out there. So please don't forget that there is a limited time for the commitment fee. So most likely, if you don't have all of it right now, between now and the end of next week, I think that's the duration you have for you to make a commitment fee. But like they say, you can pay in installment, so you could just pay what you have to secure your seat while um, um, we commence by next month. Thank you so much. Uh, you guys are amazing. Thank you. Have a great time. Bye bye.
Thank you so much, Mr. Shea. Thank you for that wonderful presentation. So, if you are interested, please kindly reach out to the admin on the host group, or you can reach out to your host so that we can push it up from there. All right, so that without wasting our time,
Sorry, please. Sorry. Lester forgot. I discovered that I think the committee gave 10 people per region. I think there was like 10 people, a slot of 10 people. I'm not sure it's open to. I just got a message, I think. Like it's not open to maybe everyone. So maybe the first 10 people, so I'm not sure. So I don't know how it is, but I just wanted to brush that in. So most likely the committee will still discuss with Genemark Institute to increase the slot. I don't know. So please just make sure you take advantage of this. Thank you.
all right, sir. Thank you so much. Just like you said, like you have to be very fast about this if you are interested. So without wasting so much of our time, we are delving into the second aspect of today's live session, and that is taking questions for day two, and this will be handled by our team lead for Gogenebio, the team lead for GSC, that's General Services and Consults, the person of Miss Motorayo in this moment. Please, we can welcome her with our emojis. You are welcome, ma.
Okay. Good morning, everyone. Can you hear me? Please, you can hear me. Just send some thumbs up in the chat. Okay, thank you very much. First of all, I would like to appreciate everybody for taking part. Yeah, we had the dream, we had the passion, we had the inspiration to bring the knowledge of things and all information to the undeserved. But we couldn't have achieved it on our own if you guys had not partnered with us. And even if the host partnered with us and people refused to register, it still would have held, right? So we want to appreciate everybody that made themselves a part of Gogenebio, the first edition. We appreciate you and we say thank you. And we sincerely hope, okay, we sincerely hope that you have learned one or two things and you have been launched out into the world of genomics and bioinformatics and you know you have gone from the stage of ignorance to now to not to learn and then you are moving on to improve yourself as far as expertise in different fields of bioinformatics is concerned. So thank you very much and we appreciate you. Okay, so without wasting much time, I will go through the two questions. Okay, so the two video was on transcriptomics. It was on transcriptomics and the platform we used was this Galaxy. Okay, so the digital video was on Youth Galaxy and it was on transcriptomics and Youth Galaxy as our tool. So we have quite a number of questions here, and I'm going to try to move through them, given the time we have. It's very short. So the first question, okay, I must also say that I saw some questions that were out of scope for the class. Yeah, and then there are some questions that were more like consultation based. So, like I was introduced earlier, I am Otunrayo, I'm also the team for Genmark Services and Contracts. At Genmark Services and Contracts, we offer services in different bioinformatics fields, and we also do consultation. So, if you have a bioinformatics project that you are interested in doing or you started and you are confused in some ways, you don't know how to move further, maybe you have some issue with your data, you have not gotten data, and you really want to just want to conceptualize your research even self. So you can consult with us. We are on LinkedIn. You can search Genmark Services and consult on LinkedIn. You will see us. So you can send a DM. You will be properly attended to and everything will be sorted. So those questions, I will not be attending to them. I will only stay within the scope of what was taught in the in this recorded session rather. So the first question here is from Chen Liren. I'm so sorry if I didn't pronounce that properly. Chen Liren is from Philippines and he said a bit of bioinformatics. How do you transfer RNA samples over long distance? Let's say Philippines to India, or do you stabilize it first as cDNA? Then pack it over dryers or you deliver the RNA as it is still as dryers. So I think one of the things that was said in the recorded session is that RNA disintegrates easily. Like at room temperature, RNA is already disintegrating. So once you get your RNA, the best thing is to transfer it back to cDNA. So even I of course there are some really comprehensive and intensive labs now that can actually produce the temperature that is able to sustain RNA. It's an evolution that is currently ongoing, but generally people convert RNA to cDNA the moment it is extracted because of disintegration temperature. So if you are now think you want to transfer it over a long distance, a lot of things can happen to your sample as it's being transferred. So you do not want to lose your sample during the transportation. So the best thing is to transfer it to DNA, to see DNA first, and then you pack it over dryers for the review. So thank you, Jenny Ren. Okay, so the next question, how do you relate the application of transcriptomics in medicine to CRISPR? So CRISPR is a gene editing technique. So the researchers, the world of genomics has really evolved now that people's gene can now be edited. So let's say a person has a mutation in their gene that is causing them to have some symptoms that are not normal. For example, somebody that has sickle cell anemia. I hope you all know that sickle cell anemia is just a single amino acid the single amino acid what's it called? It's a simple amino acid mutation. So that's what causes all the symptoms that we see for a person that has sickle cell anemia. So how do you help such a person? So CRISPR is one of the ways that are being explored to help that kind of situation, such that their gene is edited. It's not like editing in Word documents. So you remove some part and you add another thing or whatever you want to do, you can do. So that's CRISPR. So how is it related? How does transcriptomics apply to this? So transcriptomics majorly is the study of the gene being expressed per time in the cell. So how does it apply? So now let's say for example somebody's gene expression level has been analyzed to see that okay, this person has a mutation in this gene. So let me say that transcriptomics is actually how we know what gene to edit. So you cannot just blindly go and collect somebody's genome and start editing. You need to know where the problem is. You need to know which base pair it is. You need to know the codons. You need to know the chromosome. You need to know exactly where the gene is. You know now that any mistake in that kind of situation can lead to total a permanent what's it called? Permanent aberration for the person that you're trying to help. So now transcriptomics is how we even get to know, okay, this is the gene that is being affected, and then this is what we need to. So we can now apply scripture to when I edit the gene. So that's how it applies. So I can see some questions here. They are about technical issues that are being sorted, like license name and key and stuff like that. So another question is, how is the report interpreted and used? This is from Elizabeth Oluwa Family. Okay, so I'm guessing you're saying that how is reports from transcriptomics analysis being interpreted? So one of the crux of transcriptomics is what's called gene ontology and pathway enrichment. So I think the video started with gene expression, differential gene expression analysis. But in a new RNA, what's it called? RNA pathway, RNA workflow rather, it doesn't stop there. So at that point, you get a list of differential expressions. But what can you do with that list? Nothing, because it is just a list of genes that you don't even know what they mean. Yeah, genes have very weird names that you surprised at what does this mean and stuff like that. So that's why we go on to gene ontology. Gene ontology is you mapping your gene to known proteins in the body. So let's say you have a gene called BRCA. You've done your differential gene expression analysis and you realize that BRCA is unregulated in your sample. So what does BRCA do? So that's where you go for gene ontology. So from gene ontology, there are databases that you can map your gene to their what's it called, to pathways to proteins. Databases like Ensemble, databases like Grafton and all that. So that's where you map your gene to proteins, you map them to pathways, and then you see, okay, this is what this gene does. So if this gene is upregulated, if this gene is downregulated in this sample, then this is the pathway that is being affected. So that's where you start having your reports. That's where you start having interpretation for your results, and then you can start putting all that together. Of course, you must have started out with a research question. So you must have also been realizing how your result is answering your research question, and then you can start your report from there. So another question: why only they keep training using this Galaxy transcriptomics analysis? I found out that this tool was used for both transcriptomics, data analysis, and visualization. just like in, say, drug discovery and chemoinformatics, where you have computational tools used for modeling, analysis, and others, used for molecular visualization, for research publication, sharing, and collaboration. Is it not also applicable in bioinformatics, or just one particular tool does almost everything? Okay, so, but and these two we used for the video was Use Galaxy. Use Galaxy is a web-based tool that allows you to do transcriptomics workflow seamlessly. There, so what happened is Use Galaxy is like a what do I call this now? Made for you tool. So actually, so it's like a made for you tool. Like, okay, I want to make let's say I want to make a BC soup. I don't know. Okay, this is a trans country stuff. So for some people might not be familiar with a BC soup. Let's say I want to make a BC soup. So there's something called a soup mix. So the mix contains all the ingredients that you need for that egusi. So the thing you just dissolve it in water and then you put it on fire and you have your what's it called? You have your food. So instead of you going through the process of getting what's it called, getting the egusi, getting the pepper, getting the onions, getting every other thing, instead of you going through all of that, you just have one mix that contains everything, like a master mix that contains everything, and then you dissolve everything and you have those two. So that's what use Galaxy is like. So it kind of brings together all the tools you need for transcriptomics in one place. So that's why you could do analysis and visualization all together at once. But in a real transcriptomics, what's it called, workflow, there are a lot of tools that come into it. Majorly, transforming workflow start with Linux. So you use SRA kit to retrieve your data, then you do quality control and all that, and then you change your data, remove any errors and all that, and then you now get you do alignment. Once you do alignment, you have your feature count, and then you can now go to R. So in a normal standard RNA-seq analysis, R is the what's it called? It's the tool used for visualization. So you go to R, and then you analyze your research, you analyze your results, and then you have your plots. I hope everybody is familiar with the R programming languages. Okay, if you are not familiar, it's fine. So R is like a programming language that is used for statistical analysis. So you go to R, you analyze, and then you visualize, and then you can report from there. But if you want to go further, you want to train the machine learning model from your data. You can also go ahead to Python, start building models and stuff like that. So the process is actually quite long. But the reason why we use what's it called? The reason why we use this Galaxy is just for the sake of the session. The fact that you guys are beginners, and then the fact that there is not much time. But so that's why everything could be done in one instance like that. So the next question is molecular docking a bioinformatics tool? If yes, what does it entail? Yes, molecular docking is also a bioinformatics technique, I would say, technique, not a tool. So let's say the field of bioinformatics is actually a very wide one. It's a wide one that has different subunits. So molecular docking is a technique in computer-aided drug design. So now that's different from genomics. When you're doing computer-aided drug design, you're not interacting with DNA or RNA anymore. You're interacting with proteins, you're interacting with ligands, you're interacting with drugs. So what you're trying to do, you're trying to simulate the human environment, and then you're trying to get your protein to bind to some ligands to see if there's a way they can downregulate or upregulate the protein, and there can be they are druggable. So you are trying to find druggable targets, druggable phytochemicals, druggable small molecules. So that's what molecular docking is for. It's not related to genomics, but it's also a field of bioinformatics that anybody can explore if they are interested, but it's not covered in this session. So another question for the analysis part on use galaxies, we went by so fast that I couldn't keep up with the steps in real time. So we apologize for that if we could not keep up, but I think that's why the videos were made available for you guys. So you can reach out to your host to get access to the video. They have access to download the video, so they can give you the video for you to rewatch later. So analysis using visualize is good. So what research question can be used? I'm sorry. What research questions can one be asked to use this tool on? So there are a lot of things you can, there are a lot of research questions you can answer with transcriptomics. So like I said, when transcriptomics is the study of gene expression. So everything arises and falls on genes. Every physiological changes, every physiological symptoms you see, they rise and falls on the downregulation or regulations of genes. So because genes are the ones that produce proteins, and proteins are the ones that carry out many of the physiological processes that occur in the cell. So if something is not going on, then that means a gene is not being regulated properly. So there are a lot of research questions you can. So majorly I would say just try to find a symptom, find a disease condition, find a symptom, and then try to get to the root of it to realize how we can apply transcriptomics to this. So another question is, after running fast QC, I noticed several warnings and failure in the report, even after removing adapters. How do I approach cleaning and improving data quality, and which tools or methods I should use? So for data quality control in fast QC, we have fast QC is majorly the what's it called, for checking data quality. So when your data is not good, you can remove adapters using you can remove adapters using cut adapt. You can clean your data using several tools on what's it called, use Galaxy. So you can search for tools on Linux to also use um to clean your data. So that's something you can get online. We do not have time, so I'll just move on. How do transcriptomic signals differ between healthy and diseased tissue, e.g., cancer and versus normal? So, like I said earlier, everything rises and falls on genes. So how do transcriptomic signals differ? So what we do something called differential gene expression in transcriptomics. So differential gene expression is that you are trying to find the expression of genes in a particular condition in relation to the second one. So that's how it is, the difference in that's the what's it called, the signature in transcriptomics, the signal that is being measured in transcriptomics is gene expression. So you check for difference in gene expression in your heredity versus how can transcriptomics analysis guide precision medicine or drug repurposing? Okay, so precision medicine. Okay, so transcriptomics, like I said earlier, when I was talking about how the question was asked about how CRISPR is related to transcriptomics. So that's also something that can be explored for precision medicine. So you can use transcriptomics to identify which genes are being mutated, and then you can use CRISPR to edit the gene. And also you can also when you what's it called, when you build from a differential gene expression data set, you can also build a machine learning model that you can use to what's it called, to diagnose people on a disease condition. Yes, so that also helps you with precision medicine. So if you are not only considering their symptoms, you are going in depth, seeing what genes is being regulated and or down-regulated in them. You're already doing precision medicine. So and you can be very, very surprised that results can differ between human beings, even though they have the same disease condition. What transitional changes occur in response to infection, stress, or nutrient deficiency? It's still gene expression. So, and essentially, anything that is affect any what is it called? Any abnormality in the body. What transitional changes occur in response to infection, stress, or nutrient deficiency? Okay, I'm sorry. I think I didn't get the question very well. In response, if in response, so that would be that the what do you call, the release of the immune system gets into work, and we have different cells that work in that regard. So How does transcriptomic changes happen in this? Of course, transcriptomic changes can also be seen in differential gene expression. So we can see difference in gene expression in response to these conditions too. So for example, somebody that does not have infection might not be experiencing inflammation. So if you collect sample from the person and check for differential gene expression, you see that there's difference in the expression of genes that code for inflammatory proteins, all these alpha cells and is it B cells, and I'm trying to find the word antibodies. Yes, antibodies. You will see that there's a difference in the expression of that kind of gene in that person. Why? Because the person is not having infection. So compared to the person that is having infection, so you can see the changes that is occurring. So another question is how the mutation in SNPs in regulatory regions influence transcriptional output. So mutations or single nucleotide polymorphism in regulatory region will greatly affect transcriptional output. Why? Because um, regulatory regions in the genome are the regions that are regulating what gene is being regulated or is being expressed per time. Yes, so if there is a mutation in that region, what could happen is that the wrong gene can start being expressed, or the right gene might not be expressed. So that also affects the result, the transcripts that will be produced by the genome when regulation is already impaired. Can we use transcriptomics data to predict therapeutic targets or immune responses? Yes, I've already answered this. We can use transcriptomics data to predict. When you can use the transcriptomics data set to build a machine learning model, which we also have a course coming up very soon in building ML models using transcriptomics data sets. So you can use the transcriptomics data set to predict therapeutic targets for you can so if you get a gene that is strongly correlating with the disease condition, you can go ahead to map the gene to different pathways, and then from that pathway you can identify proteins that can be targeted using different therapeutic mechanisms. Which data analysis tools do you advise someone to be working with based on their experience in this genomics field? So I don't really understand this question. There are many tools that you can work on in the field of genomics. There are lots of them. So I would just say what I would advise is that start with the easy ones. Like I can touch use Galaxy now. I use Galaxy. You don't need to code when you want to use this Galaxy. So you don't have to start from the top. You can start from the bottom and then build yourself up. And really, another thing I also want to say about this is that because of the myriad of what's it called myriad of tools available in bioinformatics, it's actually very hard to get started on your own because you don't know which one to start with. So that's why it's also good for you to have a mentor. Yeah, so or you should be registered in a what's it called in an environment where you are mentored properly. So somebody should be able to ask you what are your research interests, and based on your research interests, they can recommend tools for you that you can start learning, and then you'll be able to answer questions in relation to your interests. So our advice that don't just start any tool. You what's it called? Register yourself with somebody that can guide you. And I think that virtual internship is actually a good thing for this. So if you are interested in doing but you know how to start, you can register for that virtual internship and then you'll be guided in what to learn. Because information overload is also is as bad as ignorance. Information overload is as bad as ignorance because it will cost you analysis paralysis. You know what to do because we already have too many things to do. So you need structure, and that's what those kind of trainings are for, to structure your learning. Another one is, can this training be done at a slower pace, seeing that it was stated it was for beginners, but honestly the pace has been a bit overwhelming for beginners to catch up. We apologize if that was your experience, but as I said earlier, the videos are really good, can always be watched. Okay, I saw question about IT. You want to apply for IT. in industrial training, can you apply? Can you come to Geneva? I cannot answer that here, so you can reach out to the proper quarters. My question is on CRIPSA and technology or gene editing. I understand that this technology can only work in somatic cells and non-reproductive cells, meaning the expected treatment of diseases such as sickle cell or even HIV cannot be passed on to the offspring of the patient. What hope do we have for this technology to remedy this ailment? Okay, so it's true, CRIPSA can only be applied to somatic cells and not to reproductive cells, which means that any changes that is done via CRIPSA cannot be transferred. So if a person had sickle cell anemia and was cured using CRIPSA, if the person is an SS and gets married to an SS, the person, the child of the person will still be SS, even though the mother was cured of sickle cell. Why? Because the editing only works on somatic cells. So which means the person will not have the symptoms, but they will still carry the what's it called? The gene to give birth to sickle cell people. So really, there's still much work ongoing. CRIPSA is still a field that is currently being explored. Yes, it's still being explored. So a lot of things can still happen. And you that you are asking the question, Michael Kasonde, you can also, is it Michael Kasonde? Okay, I'm saying Salota Jovu. I'm sorry about this. Okay, it's Michael Kasunde. So you that you are asking the question, you can be the one we've been waiting for. You can help us. Yes, you can help us. It's a field that is currently being explored. So anything is still possible. So we cannot say nothing is something is impossible. It is a field that is evolving every day. How to identify which one is control and which one is treated? So I'm thinking you're asking that okay, you have your data set, you're trying to identify which sample is control and which one is treated. So I think this is for the people that you are the one doing the analysis from scratch. You are the one that collected the data yourself. You have the animals, you collected the RNA yourself, and then you went for sequencing. During sequencing, you will have tagged your data, which one is control, which one is sample. So when the results comes in, so come out with that tag, so you know which one is control, which one is treatment. But if you are working with publicly available data, which you did sequence, so when you go on NCBI, there's always a session when you click the section number of the data and you go post. There's a section, the summary section. So it contains everything that was done in the research, like a summary of everything. So from that summary, you will understand the tags that are used. You understand if they say PD means Parkinson disease, so this is the treated. Then the control, you can see wild type, you can see WD, that's wild type, which means this person is control, it does not have Parkinson disease. So that's how you can, you read what's it called, you read that session, you read it. Once you read it, you will understand the data and you'll be able to work with it. Like every data analysis, it's always important to understand your data before you start anything. Okay, another question. We want to know what practical steps, learning pathways, and skill building strategies do you recommend after completing this introductory course in order to progress towards becoming a competent bioinformatician and eventually an expert in the field? I would also appreciate your advice on. Oh, so sorry for that. So I want to know what practical steps, learning pathways, and skill building strategies you recommend after completing this introductory course in order to progress towards becoming a competent bioinformatician and eventually an expert in the field. Oh, so I like this question. I feel like this person is already at the point where we envisioned everybody to be. So this is the goal of Gogenebio, to get everybody to this point. Okay, now I know what bioinformatics is, and I really want to build myself in this field. What do I do next? So this is the goal, because we understand it's not possible for you to learn genomics and bioinformatics in 3 days, but if you are exposed, the desire can be worked inside of you and you can start tracing what is necessary. So what you can do now is that identify your research interests. Okay, what research questions am I interested in answering? So is it that I want to know what are the molecular mechanisms that lead to cancer? Is it that I want to look for therapeutic strategies for Alzheimer's disease? Is it that I want to just study, just what's it called? Just help my community monitor the what's it called? The predict the outbreak of diseases. Is that what I want to do? Is it that I want to predict the outbreak of diseases in my community? Bacterial, infectious diseases. Is it I want to what's it called? Study and predict the outcome? Is it that I want to develop vaccines for these different disease conditions? What do I want to do? So when you identify that, that will help you chart a learning pathway for yourself. So you help you chart a learning pathway for yourself, and then you can start applying and registering for courses, registering for internship in that regard. Yeah, in that regard. And one of the things I also recommend is that virtual internship, which our CEO already spoke to you about. We have subsidized it very well. Yes, because our goal is not just to leave everybody hanging. We don't just want to come and talk about bioinformatics and then go, so that you still be in the same position you were before we met you. We want you to move from that position of ignorance to knowledge, and from knowledge to active learning. And the process you can do that is when you register for that virtual internship. There are a lot of there are a lot of courses available on Mixport that you can start learning from, and you can start even a research project from it. I hope I've been able to answer your question. What's the person's name? Oluwadamilola Gbolade. I hope I've answered the question. How can we fully access the information being shared? So when we fully get on with the practice, we can go back to the notes for clarity. Like I said earlier, the video will be shared. So if you need the video, please contact your host, you will get access to the videos. Another question is opportunities unlocked by this training in relation to Africa. Okay, so this is a good question. So if you are an impact person, you're an impact driven person, then you really want to make impact in your continent, in your country, in your community. Bioinformatics is one of the ways you can do that too. I was saying the other time that one of the ways you can apply bioinformatics is to predict the outbreak of diseases in a community. So you can study the different strains of a particular infectious diseases and know, okay, which this one is the most predominant in this community, and then this is what needs to be done. This is how this strain of this bacteria can be targeted and for therapeutics. So that is something you can do in relation to Africa, considering the fact that we have a lot of infectious diseases conditions. Another thing you can do is that you can raise awareness. Not everybody knows about the impact of bioinformatics. A lot of people are still held back. They have a lot of research ideas in their mind that they want to actualize, but because of money, they have not been able to proceed. But in the field of bioinformatics, you can do a simple research with publicly available data. You don't have to be the one to get the RNA seq. You don't have to be the one to get the genome. You don't have to be the one to get the DNA. You don't have to be the one to extract the protein. You just have to have access to data, which is freely available. So, and you can answer research questions. So you can start by raising awareness if you're a student, you can raise awareness amongst your colleagues and let them know about bioinformatics and how they can learn it and how they can apply it to every burning question in their mind. At least they are able to afford a full wet lab research. So it's called a dry lab research where you don't have to extract the DNA, you don't have to extract RNA, you don't have to deal with animals, you don't have to look for human samples or mouse models. You just have to have access to data and then answer your questions. So that's another thing you can do. You can raise awareness, and you yourself can carry out research. You have been informed now. You have been informed, so you can start thinking about those research questions that you have been wondering about and you don't know how to go about them. You can go on NCB and look for data. Of course, you have been after your ability skills. So that's what you can do. So I think this is the last question here. He said, I would like to thank the organizers for the excellent job. I would like to have a download training session for the days for proper digestion. Thank you. All the videos will be available. We made them downloadable. So we made them downloadable, but they are not resellable. Please let us understand that they are downloadable, you have access to them, you can watch them anytime you want, but they are not resellable. Please, you don't have copyrights over them. They are only for educational purposes. So you also have friends that are also interested in learning, but you can share it with them, let them learn. The goal is for everybody to learn. So thank you very much. I think this will be all for the two questions. So thank you. We appreciate your presence and we hope you've learned one or two.
All right. Thank you so much, Ms. Muswayo, for that impressive answer. Thank you so much. I mean, that actually cleared all the questions and the clarifications about some of the things meant for the one and the two of Golden File. And so we've come to the end of Golden File. We've come to the end of Golden File. I know that this has been an impactful session with everyone all over the world. And I would like to inform everyone that for people that are interested in the virtual internship or in some of our short courses and programs, um, a, a contact will be sent to the group to, if you want, if you are interested, so you can just send a message to the number, and then it's one of the general hotline, and then you will be attended to as soon as possible. So if you are interested, so please send the message, contact us, and you can also contact some of the admins on the group. You can contact some of your hosts, some of the people that helped you to come for this, to be able to attend this program so that we can do so many things together. So we are ending the session now. Thank you so much. Thank you to all the hosts that pushed this program to success, to being a reality. Thank you so much to all the participants worldwide. We are so grateful that you were able to learn one or two things. You were trained. You got to understand more about bioinformatics and transcriptomics in a deeper way. Thank you so much for joining. And I would like to appreciate Genoma for this as well. I would like to appreciate the director of Genoma Institute, as well as Ms. Mosrayo for their time, for their efforts, for their cooperation to this vision. We say a big thank you. Thank you for your hard work. Thank you for your commitment. And then to all the hosts and participants, thank you. See you again. Bye, everybody. So we can end the session now, officially. Bye.